The overall goal of the following experiment is to deliver macromolecules into live cells using dimer fluorescent TAT or dfTAT, a reagent that can penetrate cells very efficiently without damaging them. This is achieved by first generating and purifying the delivery agent dfTAT. As a second step, dfTAT is incubated with cells for 1 hour at 37 degrees Celsius in the presence of the macromolecule cargo of interest. dfTAT induces the uptake of the cargo inside endocytic vesicles. Vesicles mature into early endosomes, finally reaching the late endosome stage, where dfTAT is able to release the cargo into the cytosolic space of cells. Next, the cells are imaged by fluorescence microscopy in order to assess the delivery efficiency of dTAT and the cargo of interest. The results show that dfTAT can enter cells with high efficiency and no observable cytotoxicity. dfTAT can deliver macromolecules of different sizes into the cytosolic space of cells, as can be observed by fluorescence microscopy.
The main advantage of this technique over existing cellular delivery approaches is that our delivery agent has a high delivery efficiency with no observable negative impact on cellular physiology. In addition, it is easy to use as it requires only a simple co-incubation step.
To begin fTAT synthesis,